Good afternoon, and welcome to Plenary 2. Please welcome Senior Vice President for Clinical Research of the Cystic Fibrosis Foundation, Dr. J.P. Clancy.
Woo! That was awesome. Give it up for the band, Rio. Oh my gosh. You guys as excited I am? I tell you, you are in for a treat. I saw it this morning. It's good, all right? So just be ready, be forewarned, and be ready. So let's get this thing started, shall we? All right, well, welcome friends and colleagues in attendance and online to the second plenary session of the 2022 North American CF Conference. For those who don't know me, my name is J.P. Clancy. Just said, I'm the Senior Vice President for Clinical Research for the CF Foundation. And it is so exciting to gather here in person this year to review and share the latest and most exciting insights in CF care and research. The tremendous advances that are being realized today in CF build upon the work and commitment of many talented people who helped pave the way for our current success. Today, we remember one of those past leaders, Dr. Richard Talamo, and present this prestigious award named in his honor to one of the giants of pediatric CF research and care. After graduating cum laude from Harvard and earning his MD from Boston University, Dr. Richard Talamo pursued a career in pediatrics and became an NIH clinical fellow in the laboratory of Dr. Paul de Saint-Ignace, focused on CF. While a professor of pediatrics at Johns Hopkins, Dr. Talamo became deeply committed to the mission of the CF Foundation and served in many tangible ways, including on the Medical Advisory Committee and the Board of Trustees. During this time, he helped raise awareness in CF and encouraged researchers to focus on this disease. Dr. Talamo returned to Boston in 1980 as the chairman of pediatrics at Tufts University. But sadly, this brilliant and caring man died at the age of only 47. The Richard C. Talamo Distinguished Clinical Achievement Award recognizes individuals who have spent their careers researching and caring for people with CF and whose contributions have significantly impacted the disease. This year's recipient had a highly distinguished career in CF for over 40 years with huge impacts that cut across care, training, and research. This year's recipient is Bonnie Ramsey, MD, professor in the Department of Pediatrics at the University of Washington and Seattle Children's Hospital. I'm warning you, Bonnie, there's more clapping to come. I'm just saying, all right? Be aware. All right. So just to give you a brief background of Bonnie's career, trying to pull it into one small space, Dr. Ramsey completed her Bachelor of Science in Biology at Stanford University and then followed that with medical school and residency at Harvard. She went on to be senior resident at the University of Washington, where she subsequently started her illustrious career as faculty at the university and in the Department of Pediatrics. Bonnie has been at the forefront of cystic fibrosis clinical research for decades, with deep interests in understanding and treating CF infections, improving pediatric CF outcomes, and advancing symptomatic and CFTR-based therapies. After leading research efforts that resulted in FDA approval of inhaled tobramycin for people with CF with chronic Pseudomonas infection, Dr. Ramsey had the radical idea and audacity of establishing a CF clinical trials network to advance new therapies. She met with then CF Foundation CEO Bob Bell, and what emerged from that conversation was the Therapeutic Development Network, or TDN. Dr. Ramsey guided this fledgling collection of 8 independent academic research programs into a robust, collaborative, and efficient network of over 90 sites. In those early days, there were few, if any, industry sponsors interested in developing new CF therapies. But because of Bonnie's leadership and her development of the network's capabilities, industry interest in CF and new therapies, it grew, and building upon success that ultimately became the pipeline that we see today. Numerous therapies that are available to CF patients today have passed through the doors of the TDN, including enzymes, inhaled antibiotics, mucus hydrators, anti-inflammatories, and approval of ivacaftor as we entered the modulator era. She also guided the TDN through the genetic studies conducted in the early 2000s. And while these trials were unsuccessful, they helped provide critical insight and are now an important stepping off point for the path to a cure in numerous genetic therapies entering clinical trials, as discussed yesterday. Bonnie has been recognized far and wide for her enormous contributions that are too numerous to summarize here. However, what is not captured in these accolades is her critical role model status for women in science, her commitment to mentoring young faculty and scientists, and her love and dedication of the CF patients that she cared for. I personally experienced the Bonnie effect when a patient of mine temporarily relocated to Seattle, and upon her return, I was regularly reminded that Dr. Ramsey probably had an answer to any question that arose or confusion that I might have. But enough from me. Let's hear from some of Bonnie's peers as they pay some tribute to her and her career that has truly transformed CF.
We live in a time when it's easy to take for granted things that seem as if they've always been there. And I want to honor Bonnie Ramsey by reminding us of what didn't exist before she brought her extraordinary talents to help advance CF treatment. You know, before Bonnie envisioned the TDN, research was done at individual centers or maybe with collaboration between colleagues. There was no assessment of standardized outcomes or issues of study design or consent, and there was general disdain for working with pharmaceutical companies. Bonnie is a critical thinker. She has incredible organizational capacity. She has the vision of the possible, and it was those qualities that helped her develop the TDN.
She always seems to have been a fierce advocate and has devoted her energy to all the things that she's had a passion for, her patients, the CF community, research in CF, and clearly her dedication to forming a network to help really accelerate development of new drugs for people with CF. But Bonnie also brought along research coordinators in a way that they'd never been appreciated before. Her partnership with Judy Williams really was the model for the way that investigators and research coordinators have functioned in the network, and something that we bring up repeatedly as new teams come on board. Her impact has really been so phenomenal.
One particular memory that I have of Bonnie is from the very first meeting in 1998 of the CF Clinical Trial Network that we now call the TDN. And it's where Bonnie laid out her vision for why it was essential that we would form a network, work together to do CF clinical trials, and that she would lead a center that would coordinate it all. What she talked about that day is exactly what has happened. The TDN's grown dramatically in size, and most importantly, it's conducted every important clinical trial for the medicines that now are transforming cystic fibrosis.
I remember the early intervention study with antibiotics that she did. And then, of course, helping with the CFTR modulators, both the discovery and the development, and really helping to bring forward lots and lots of experts into CF who otherwise may have chosen other paths.
30 years ago, I used to be asked, why did you go into CF? And I went into CF because of the families and the patients and their commitment and their strength in facing adversity really was an inspiration to me. So I feel very, very pleased that I've been able to give back a little bit to the families.
Bonnie is an amazing person. I think she's a once in a generation, or even less often. She's incredibly motivational. She's incredibly inspiring. She's visionary. She always sees the big picture, but also sees the details. And she's an amazing leader. She gets stuff done.
She is selfless. That has turned her into a leader that we all follow. She always thinks of not herself, but the patients and their families. And that has given her perspective in life that has always taken us and grounded us in thinking about how we advance CF. It has created an eternal bond to everyone who's worked with her.
On behalf of the greater CF community of CF families, care providers, researchers, and beyond, I have the honor of thanking you and for all that you have done for CF, Bonnie. Thank you.
Thank you to everybody in this room and to all the patients worldwide. I am just so honored and overwhelmed. I love you guys. Thank you.
Well, I do have one last surprise. And Bonnie, I just want to let you know this will be the last time that the Talamo Award is going to be presented, because from this day forward, this is going to be known as the Bonnie Ramsey and Richard Talamo Distinguished Clinical Achievement Award. Nothing to say, but thank you. Now on to the show. So the second plenary presentation will focus on the important role that all of us will play to bring new therapies and ultimately a cure to CF. CFTR modulators have led to transformational change for many people with CF, but our work is so far from done. Many people are not candidates or not able to take modulators. Some don't receive benefit, others have side effects. Importantly, new symptomatic therapies are needed to treat mucus infections, inflammation in the lungs, to treat diabetes, liver disease, GI symptoms, and nutrition, just to name a few. And finally, developing a cure for CF will need new ways of conducting clinical research with new questions to answer about studies of genetic-based therapies. To take us on this journey, I am delighted to introduce the two speakers for the second plenary. The first speaker is Dr. Deepika Polineni, who is an associate professor and has recently been named the CF Center Director at Washington University in St. Louis. Her part of the plenary will give us a glimpse of where we are today in new and novel therapies for CF. During her fellowship at Chapel Hill, Dr. Polineni met Dr. Michael Knowles, an adult pulmonologist and world-class CF researcher, well regarded as one of the leaders in thinking about understanding the basic defects in CF. Not being a fool, Dr. Knowles recognized her immense talent and helped to grow her research expertise in CF, leading a research team evaluating airway epithelial transcriptomics, enrolled in a large multinational study of CF genetic modifiers. While at the University of Kansas, Dr. Polineni continued to grow her basic research program and developed into an international CF leader and clinical trialist. She has been intimately involved in multiple pivotal trials of new therapies over the past decade and is a dedicated mentor of junior scientists and provides national leadership for women in science. Dr. Polineni will then be joined by Dr. Nicole Mayer-Hamblett, a professor of pediatrics and biostatistics and co-executive director of the Therapeutic Development Network Coordinating Center in Seattle and the University of Washington. Nicole's part of the plenary will give us a glimpse of the future and the important role that the entire CF community will play in reaching our cure. After joining the TDN in the late 1990s, incidentally, under Dr. Ramsey's leadership, Dr. Mayer-Hamblett's role and responsibilities in the greater CF research universe have steadily grown. She has become intimately involved in the design and conduct of nearly every large CFF and NIH-funded clinical trial conducted by the TDN over the past 2 decades. Nicole has been at the table numerous times discussing CF clinical trial design and conduct with the FDA, with partners in the NIH, and with the CF researchers globally. She is a key liaison with industry partners seeking to develop new and novel therapies for CF, which now extends to novel genetic-based therapies that are at our doorstep. Nicole is also a critical thought leader in the development of large studies seeking to better understand the care of CF management in this new era of highly effective modulators, including such blockbuster studies as GOAL, PROMISE, CHECK-SC, and SIMPLIFY. I can think of no better qualified colleagues than Drs. Polineni and Mayer-Hamblett to take you on this journey. And between them, I can guarantee you that you will have new appreciation and perspectives about the exciting territory that we are entering. So Deepika, the floor is yours.
Thank you, Dr. Clancy. I want to thank Dr. J.P. Clancy, Dr. Mike Boyle, the Cystic Fibrosis Foundation for the opportunity to articulate this roadmap that's in front of us today. It's such a blessing to get to do this. And I really also want to say that this presentation is truly a reflection of all of the people that contributed their thoughts and their data towards this discussion today, so thank you to all of you. These are my disclosures, and that was one heck of a page turner of a pandemic. So between all the twists and turns and everything else that could have occurred, we had Francis Collins guiding us through it and leading us through a global pandemic. The last time that we were together in a room, Dr. Collins was leading us together in song. And in fact, it was the song Dare to Dream, which is up in the top right corner, which he wrote for people with CF. And I draw your attention there because there are 3 pictures there today that I'm privileged to get to discuss with you. These are iconic images in cystic fibrosis, and I've changed the way that I look at them and think about them. And I hope that after this, you might too. The first of these 3 images was on the front page of the Washington Post in 2019 when the news of the FDA approval of Trikafta broke. This is the most recently approved CFTR modulator, and there is no question that this was historic. The title reads, "Long-awaited cystic fibrosis drug could turn deadly disease into a manageable condition." But I think this is still a little inaccurate, and here's why. Because what we know is that ivacaftor was actually approved a full decade ago this year. In 2012, 10 years prior, we had patients starting on ivacaftor, and today we continue to learn from research participants, patients and families who have lended their experiences and stories to us on a highly effective CFTR modulator. And that has also guided our knowledge of what we need to know in terms of moving forward in an era of Trikafta for people with CF. And what's more, this entire timeline shows in color coordination here how therapies along the way have really been moving the dial on turning this deadly disease into a manageable condition for people with CF for so long now as well. And these treatments are really aimed at targeting every aspect of what you see here on this familiar wheel of CF pathophysiology, and every endeavor is still made today to continue creating treatments that will help target these portions of what happens in cystic fibrosis. And they are moving on with great success. So I'm happy to share these data with you today, courtesy of Vertex Pharmaceuticals. These are the data that are representing the 144-week interim analysis in elexacaftor, tezacaftor, ivacaftor, or Trikafta in people with at least 1 delta F508 mutation and a second minimal function mutation. These are the secondary endpoints that I'm going to discuss today, and I think it's important to call out that Dr. Corey Daines will be discussing the full results of the study in the workshop following this plenary. But here you can see that following the pivotal trial, which is shown on the right and shaded in white, patients who were on placebo were rolled onto the drug therapy, and you see the sweat chloride levels plummet for those gray circles. Not only that, you see that continuing out to 144 weeks, and so it certainly looks like these effects are sustainable. What's more, those historic gains in lung function also appear to be sustainable as well. At 144 weeks in this open label extension study, we see that FEV1 continues to remain high. And further, this is a trend of stability that we did not see to this degree even with ivacaftor in the past at this point in time. So again, I think that it's safe to say based on these analyses that Trikafta is here to stay, and I encourage people to visit Dr. Daines' workshop to learn further about this study. Also, what we learned at this conference is very exciting, that as we continue to try to find CFTR modulators that can be used in younger age ranges, these data were very promising to see and have actually supported the application from Vertex Pharmaceuticals of a new drug application to the Food and Drug Administration for Trikafta in children 2 to 5 years old just yesterday. These are the secondary endpoints for this open label. These are also secondary endpoints in this open label extension study following the pharmacokinetic analysis. And this is representative of 71 children. And you can see again that the sweat chloride levels are reduced and appear to be staying there out at 24 weeks. What's further, lung function is improved as reflected by the lung clearance index that's shown here. So this is in fact the number of cycles that it takes to turn over or expel gas from the lungs. So a lower number is in fact better. And this is showing that reduction after treatment and persistence at week 24. So I know that I'm showing you a blank slide. This is not an error, but I wanted to translate this first because you're going to see lots of colored lines. What we have that I get to show you next is an independently funded study from the Cystic Fibrosis Foundation Therapeutic Development Network that many of you across this room contributed to. This study was called PROMISE and led by Dr. Dave Nichols and Steve Rowe, and these are actually the 6-month health effects of elexacaftor, tezacaftor, ivacaftor, or Trikafta that I get to show you here. Patients in this study were either taking no prior modulator or a true 2-drug modulator or ivacaftor. And after 6 months, what we see in terms of the context that you might hear in clinic or in a clinical setting is that sweat chloride levels were dropping to below 60 millimoles per liter. That is in fact the diagnostic threshold of what we think of for cystic fibrosis. And what's more, for patients who were taking ivacaftor already and were put onto Trikafta, their sweat chloride levels were reduced to thresholds below 30, and that is the threshold that we think of for a CFTR carrier, or someone with just one copy of a mutation in their CFTR gene, perhaps a parent of someone living with CF. We see these kinds of improvements in lung function as well. FEV1 has unequivocally risen in the data in this study into thresholds of what we would call normal lung function. And Dr. Felix Ratchen is going to be discussing the pediatric PROMISE results, as these results are actually in patients 12 years and older. So the pediatric PROMISE results will be discussed following the session as well. Investigators in the United States, across the European Clinical Trial Network, and more, are searching for ways to have CFTR modulators like Trikafta and more able to access patients with rare mutations so that we can make the most of extending these types of health benefits to everyone who can receive them with CF. So we've heard about theratyping before, and I think it's very exciting to say that we have now studies that are being conducted in the United States for mutations such as N1303K and other rare mutations, where we see that investigators are using induced pluripotent stem cells or even nasal brushes that are fully differentiated then to airway epithelial cells and tested in the dish to see whether short circuit current changes after exposure to CFTR modulators. Similarly, in the European Clinical Trials Network with studies like HIT-CF, we're seeing the use of intestinal organoids and seeing whether there is a swelling response in terms of treatment with CFTR modulators. But then this is taken one step further to understand whether or not we're seeing the correlation of in vitro findings to findings that are clinical outcome measures that we would see in the clinic, such as FEV1 or sweat chloride. And it is exciting to say, as stated in the abstract 606, that some of these findings have already resulted in the approval of therapy for people with CF. It's through measures like these that we heard about these numbers yesterday, through the reach of investigators that are pushing every limit to see where we can take these therapies. We now know that we can reach up to 94% of people with CF with a highly effective CFTR modulator like Trikafta, and there are still more under research. Poster 662 suggests just one of those that is currently under investigation. So I think it's easy to say that CF research is far from over. And why is that? That's also because of the 6% of people with nonsense or other rare mutations that are currently ineligible for CFTR modulators, and people who simply cannot tolerate modulator therapy, that we continue to press forward and press on in this research. And we heard about this a little bit yesterday, and I'll summarize again today. To me, what is extraordinary about CF is actually the true diversity of CF. Now we know that there are about 40,000 people in the United States and more than 100,000 worldwide, and that CF can occur in any race and ethnicity. This is resulting because of mutations in the CFTR gene, and despite this being a monogenic disorder, the disease severity and manifestations that we see can vary so much. And that is due only in part because of the many variants that we see in the CFTR gene. There are more than 2,000 variants that have been characterized, and the majority of these are disease-causing. So it is actually, that is exactly what disrupts this process that you see here. You see the CFTR gene on chromosome 7 that is first transcribed to mRNA, and then translated with post-translational modifications as well to CFTR protein, all to configure this beautiful 3D structure, this ribbon format that we have here of the CFTR protein in its perfect condition, which functions to gate normally at the cell membrane and transport chloride. But what we also see is that the CFTR gene variants represented in this pie graph have a substantial portion that will create a disruption to this process that will not even afford a protein that a modulator can act upon. And it is for these folks especially that we keep looking forward. This is the second of the 3 images that I called out to you, and I want to take a second to explain why I think this is iconic. I know there are some of you in the room saying, Deepika, we've seen 3D ribbon structures of CFTR before. But this review article was incredibly poignant and well done. So if you have an opportunity to read this, this was published in 2020. And what really struck me was that this author wrote, in memoriam, the author dedicates this review article to his youngest brother, Nilo Lopez Pacheco, who fought against CF for 26.5 years. And when I read that, it truly struck me because I can't think of anything that's more emblematic to the fervor and dedication and commitment of this research and investigative community and people working across this field whose lives are directly impacted by cystic fibrosis, some of whom I know, so thank you for your work. So we take from that this opportunity to look forward at what happens when we can't have protein that can be treated with the CFTR modulator. And for that, we have to take a look backwards. So yesterday we heard about the path to a cure and opportunities to try to treat these defects at the RNA level, but I'm going to talk a little bit about what happens if we go just a touch further back to the DNA level, such as with gene delivery or gene editing. And I think what has to be emphasized is that we'll also be covering the fact that no one is stopping to look at the multi-organ manifestations that really constitute this disease and trying to work at treatments that relieve the symptoms and lessen its burden. So these groundbreaking data were shared in Symposium 1 of this conference yesterday. And if you missed it, I would highly encourage you to find the recording and hold on to it. These are the interim results of the 4D710 aerosolized adeno-associated virus genetic medicine for CF lung disease. This is a phase 1/2 trial, and cohort 1 successfully enrolled 3 patients who received a single dose of 4D710, which has thus far been shown to be safe and well tolerated. But what I think is remarkable is that 100% of lung samples that were obtained showed the modified CFTR transgene expression modified for the ability to detect it on assays such as these. So in the bottom right corner, you see a right endobronchial lower lobe brushing, and this is giving a broad sheet of cells, and those dark areas that you see are actually signal that are showing that the RNA is there from the CFTR gene delivery, so it's gone through that first step. And it's a signal from in situ hybridization. The magnified signals in the center actually show at 40x that the signal is being picked up in airway epithelial cells, and just lower at 80x, there's suggestion here even of secretory cells. The bar graph on the right shows that in these 3 participants in both the left and the right lung, I'm sorry, 2 for the left lung, the second did not have a sample obtained there, we can see that there is a spread of 36 to 40% of epithelial cells showing this signal. This was a safety study, so the dose was started at a low dose to understand the safety and tolerability of this drug, and while it is far too early to try to draw any inference of what the efficacy of this might mean, the efficiency of this kind of transduction and gene expression is truly promising and something that we have to pause to take note of at this conference because this is a milestone. There are more than 5 clinical genetic therapy trials that are planned or under review in 2022, and as we've just noted, 4DMT is currently enrolling, but you can see here just a small representation of these trials that are waiting to get out of the gate, and Dr. Rowe mentioned yesterday that there are 60 under current funding and discussion in various stages of planning with the Cystic Fibrosis Foundation. So to fit these many trials with one mission, Dr. Nicole Mayer-Hamblett will be speaking next about clinical trial strategy to make sure that we can meet the needs of all. But I want to introduce you to one such person who is waiting on these types of therapies.
My name is Trey Newman. I have cystic fibrosis. I have 2 rare mutations where I can't take modulators. What's up, my boy?
What's up, babe?
Hey, you ready to do this thing?
Yes, sir.
I am a social worker for homeless services, and I act part-time. I do plays, movies, skits, parodies. I like to play basketball when it's weather permitting. I also love playing basketball on Xbox as well. If I can get paid for it, that'd be great. But there are a lot of people who don't have full-time jobs, so they have that upper hand on me. I'm gonna give it to you. Okay, ready? Exercising, dealing with CF. As a kid, it was easier. My lung function was better, and I played basketball a lot more than I do now. But as you get older, the buildup of mucus, that makes it harder to play or exercise for longer periods without having to stop or without, having to cough. Or if I'm walking long distances, give me a break. I need to catch my breath for a second. And I'm only 28, so I'm used to it. But other people, not so much. Other than my lungs, CF affects my kidneys. I have to do nasal rinses. I also have CF-related diabetes, a vitamin D deficiency, as well as a multitude of infections. Because of infections, I tend to go into the hospital 3 to 4 times a year. When it comes to clinical trials, I have participated in a few targeting those who had complications with CF. I feel like a lot of people are like, well, why would you do that? Don't you feel like a lab rat? And I look at it as, well, what can go wrong or what has gone wrong with people who have participated before me? What has gone right with people that have participated before me? And I look at those very closely, and usually the pros outweigh the cons. I feel like it's building blocks to help the CF community. I think it's important to have clinical trials for genetic therapies because I feel like a lot of times people like me or those who can't take the modulators, I feel like we're like, oh well, we'll just keep doing what we do, what we have been doing. I feel like it gives us hope. It gives us something to look forward to in the future. In the perfect world, my hopes and my dreams would be a cure for cystic fibrosis. If there's no cure, I hope for those therapies and better medicine and less workload for CFers. That would be perfect.
He's every bit as amazing as he seemed there. So Trey said, in a perfect world, I want a cure. And if I can't have a cure, then I want other treatments that will continue to help make this easier. And I know that that's what everyone that is working across this pipeline in, and I have the privilege of selecting some of those to discuss with you today. So in the anti-inflammatory pipeline, it is exciting to tell you that following this session, Dr. Larry Lands will present the results of the Applaud Phase 2 study. This is a pro-resolution drug candidate, LAU7B, that was tested in adults with cystic fibrosis. But importantly, it has to be emphasized that this study was completed despite the course of the pandemic and pivoting to the onset of Trikafta. So that is a significant accomplishment. And investigators are looking at novel ways to consider looking at the outcomes of inflammation with different biomarkers, or how we're looking at lung function decline, particularly as the landscape of pulmonary exacerbations changes with Trikafta. This is critically important because in 2020, we still continue to learn that even on long-term ivacaftor therapy, there is no conclusive evidence to date to suggest that highly effective CFTR modulators are really significantly reducing inflammation. And when we consider the course of lung function decline after 5.5 years of Trikafta in the GOAL study, we see that pediatric patients, or those with the most mild lung disease, actually have the steeper rate of decline. So we think maybe if we can get in front of this a little bit, in front of both infection and inflammation, we may be able to help stave this off. And the anti-infective pipeline is incredibly robust and busy because of that as well. There's no way I could cover all of this, but Trey mentioned that one of the bacteria that he has is nontuberculous mycobacteria, and another is Pseudomonas. So one of the novel posters that is available at the conference this year is looking at the application of inhaled gallium, which we've seen treatments with IV gallium for Pseudomonas before, but subjects are currently enrolling in a study of inhaled gallium for treatment against Pseudomonas. And what's more, investigators are looking at applications of IV gallium to treat nontuberculous mycobacteria. This is a difficult bacterial infection that affects up to 20% of people with cystic fibrosis, and I can tell you that the treatment regimens are sometimes as challenging as the bacterial infection itself. And that's why it's so important that investigators are continuing to look at novel ways to try to combat this. Some of this involves phage therapy. So this paper in Cell was published earlier this year in the summer by Jerry Nick and colleagues, and they in fact described a case of a patient who had Mycobacterium abscessus infection, a particularly challenging infection which can in fact bar a patient from being able to receive a lung transplantation. This patient underwent bacteriophage selection and was treated with a cocktail of phage therapy and monitored over the course of a year with careful monitoring of biomarkers for response. And this patient went on to successfully receive a lung transplantation, and 4 months after which did not show evidence of abscessus recurring. So investigators like Dr. Nick and others are looking at those biomarkers and patient selection strategies to formulate a clinical trial and try to understand how we can look at phage therapy and broader applications outside potentially of this type of emergent use. And Trey also supported the need for mucociliary clearing agents with his continued cough. In this conference, we do have the results of the, phase 1 study of ETD-002 that was presented in poster 180, showing that this TMEM16A potentiator, or alternative chloride channel potentiator, that is really leveraging other channels that could maybe overcome the defect in what's happening in CFTR with their ion transport, has shown that it has successfully and safely enrolled healthy volunteers. And in 2020, we had the FDA approval of inhaled mannitol. But what do these types of agents mean in a setting of Trikafta? Well, for that, we have to kind of look back to the PROMISE study, which I mentioned earlier. This sub-study of mucociliary clearance was conducted and shows the percentage of clearance from the whole lung of these radionucleotide-labeled particles that are being exhaled from the lungs over time with cough assistance early on in this time curve. But what you can see from the lower portion of the curve that was taken at baseline is that 1 month later in the blue line, subjects clearly have a higher level of mucociliary clearance from the whole lung. Can we build on this to create even better mucociliary clearance for patients? So what you see here with the asterisk is the actual improvement in mucociliary clearance for patients with moderate to severe lung disease. This was taken for patients with FEV1 of 70% or less. And what's hypothesized here is the percent change that we might be able to see by adding inhaled mannitol onto that treatment regimen. Investigators from UNC will study this question in 22 subjects in a multi-center trial starting in 2023. This is actually one of the best moments for me to get to present to you, because one of the most challenging conversations that I have as a physician with patients is discussing the onset of cystic fibrosis-related diabetes. We know that CF affects so many organs, including the pancreas. And earlier this fall in September, the findings of a pivotal trial of a multi-center randomized study of a bionic pancreas and type 1 diabetes, which is not the same, but has some overlapping properties with CF-related diabetes, were published in the New England Journal of Medicine. And this is actually the study that has been taken to the FDA to move towards approval for the bionic pancreas use. And it is exciting to say that Dr. Melissa Putman and colleagues have led a crossover trial comparing 2 weeks of insulin-only bionic pancreas to 2 weeks of usual diabetes care in 20 adults with cystic fibrosis-related diabetes. What I want you to try to look at is the total time that you're seeing over 24 hours in this continuous glucose monitoring between the 2 red dotted lines versus the total amount of space that is between the 2 blue dotted lines. And what you can see is that there is significantly more time of that space that is within range of these black horizontal lines for the subjects that were treated with the bionic pancreas versus those in usual care. I think what's further exciting is that the degree of the difference of these results is similar to what was seen in the New England Journal reported trial. Dr. Putman and colleagues are taking this forward into a broader study to see whether this can be applied further in cystic fibrosis. These are just a few of the different aspects across the treatment pipeline, but we know that there have been so many more over time. So let's take a look at where the pipeline has brought us. Now, someone told me you can't see any faces from up here, but when you can see even Mike Boyle's face amazed by that story, it's pretty great to see. That's a lot of shots on goal across a lot of breadth. And you have to ask yourself, why? And I'm so honored to get to show you why, from my perspective. We continue to have this many shots on goal across this much breadth to make sure that we're meeting the needs of everyone. This is the last graph I'll show you, and it's one that's very special to me because the late Peter Dury is one of the most kind and incredibly dedicated physician scientists that I could have gotten to know in cystic fibrosis. And it's because of him that I learned through looking at 1,357 Canadian patients with cystic fibrosis who had 2 copies of a CFTR mutation that was termed pancreatic insufficient or would have been severe, or considered to be severe, that I learned that you can see this kind of spread of lung disease severity, even though you should relatively be able to control for how much their CFTR is functioning. This simple plot alone tells me so much. It tells me that every one of these patients is unique, but also it dispels one more thought that I had, and that brings me to my third and final picture that I'll discuss. CF is more than CFTR in this representation, but also in the way that we look at all of our patients. The heterogeneity that we see is so reflective of their uniqueness, the types of disease manifestations that they have, the modifier effects that are occurring. We work together to cover this pipeline so that we can carefully continue to tune and meet the precision needs of therapy for every person with cystic fibrosis, because we know that it can be as different as siblings are with each other. To be able to treat everyone, it's going to take everyone. And that is why I firmly believe that there is more work to be done. So 2-3 years ago, Dr. Francis Collins asked us to dare to dream, and I won't read to you the entire quote up in the top right, but I will actually ask you to read it, because I really do feel that it reflects all of you, and also because I can't think of a better way to introduce or describe my next colleague and speaker who's coming up here, Dr. Nicole Mayer-Hamblett. And I know that Dr. Mayer-Hamblett and I not only agree that it's not done until it's done, it's until it's done for everyone.
Thank you, Dr. Polineni, for the kind introduction and for sharing the incredible pipeline of new therapies that we are currently progressing. I, too, am so humbled to be here today. You know, when I started my career as a statistician more than 20 years ago, I want to confess to all of you that I thought clinical trial strategy was all about statistics. I started my very first job at the Therapeutics Development Network with very little experience. However, I did bring this book from school with me. I was so excited and prepared and ready to do any sample size calculation that was necessary, until I did my first one, which those of you, if you're out there from Baylor, you may remember this one, but I proudly announced that we needed over 1,000 trial participants. Now, my mentor, Dr. Bonnie Ramsey, she was so kind and patient. She quietly pulled me aside. I'm sure she does not remember this, but she said, Nicole, you know, no CF trial had ever or probably could ever be that large if we are ever going to get these therapies out to our patients. I learned more from Bonnie than I did any stats class, as there's nothing textbook about designing clinical trials for a rare disease such as CF. So just as I did 20 years ago, we are also going to put this book to the side today. Don't get me wrong, I love this book. But clinical trial strategy is fueled instead by an indescribable magic that happens when we all partner together to move this pipeline forward. Let me show you why we're going to need so much of that magic. Dr. Polineni just showed you a great video clip of the transformation of our pipeline over the years. I want to show you this transformation from a slightly different perspective. In the very early days of our pipeline, we were, for the most part, designing clinical trials one by one to tackle conditions that the majority of our CF population were affected by, such as Pseudomonas aeruginosa, mucus production, and inflammation. Well, fast forward to today, and we've moved well beyond a one trial at a time strategy. Thanks to the foundational work of our clinical trial networks around the globe, our trial pipeline is working hard to include trials that will collectively capture every mutation as we pursue disease-modifying therapies for all. Trials for every symptom or condition associated with CF, no matter how prevalent and whether you are on or off a modulator therapy. And trials for every priority raised by our CF community. This is the pipeline we are working towards to meet our mission, and it's the beautiful vantage point that I get to see our pipeline every day. Now, there's a lot going on in this graphic. It's very colorful. It's not quite complete. But it's the only way that I can visualize for you that when we talk about clinical trial strategy, it is not from one trial at a time, but is with the vision of how we are going to move all of these trials through the pipeline successfully at the very same time. Fortunately, it's not hard to find synergies combined with just a little bit of that magic to get this work done. So today, I am going to break this down a bit and highlight synergistic strategies within each of these types of trials, hoping that you will keep in mind that we will share these strategies across our pipeline. So let's start with trials to ensure that every individual with CF, in particular those currently ineligible for CFTR modulators, will someday benefit from a disease-modifying therapy. As we've heard from our prior plenary speakers, there are many early phase trials planned or underway that are in the critical path towards developing safe and effective genetic therapies. I want to take a moment to acknowledge early phase trials are more intensive than later phase trials, longer and more frequent study visits. But it's important to realize that the information that we gain from even a single study visit at this phase of development is helping us to move this entire pipeline of new therapies forward. So despite being more intensive, these early phase trials are in the critical path, and we are faced with progressing these trials in a relatively small proportion of our patient population. Now, the Nicole 20 years ago, I'll admit, would be super excited to jump into study design here. But we need to keep that book aside for a moment, as we really need to begin with starting to listen to our CF community. In a recent international survey, we see the dichotomy that the majority of people with CF not benefiting from modulators are interested in trial participation. But when asked why they had not or would not participate in a clinical trial, there were multiple reasons, including both lack of opportunity and barriers that range from significant logistical concerns to concerns about the impact of a trial on one's health. This is why our trial strategy must begin with increasing opportunity and reducing barriers to trial participation. Now, this is a very tall mountain of opportunity, and we must ensure that everyone can make it to the top, which is why our first milepost on our journey is to reduce barriers to ensure equitable opportunity. And this is where we'll truly take the work of everyone in this room today to remove the individual to policy level barriers that stand in the way of trust and opportunity as we work to engage our entire community, and in particular those historically underrepresented in research. This will take everything from increasing the diversity of our clinical and research teams, all the way to ensuring the transparency and reporting of study results by, for instance, race and ethnicity, and getting those results back out to our community. It is each and every one of our individual commitments and contributions that will get us to this first milepost. There are many steps along on this mountain, and along the way, we will be relying more than ever on partnerships between people with CF and their care providers to actively discuss research opportunities, not only focusing on those opportunities that are close to home, but those that provide the necessary compensation for time and travel to research sites across our clinical trial networks, realizing that sometimes the best opportunity for a participant or for someone may not necessarily be close to home. And we will need to provide everyone with the tools and resources to equip everyone to transparently discuss the risks and benefits of trial participation. It will take everyone to get everyone to the top of this mountain. And we will be working across our pipeline to increase opportunity and reduce barriers to trial participation. And yet, even when we create this opportunity, we must still be mindful that our genetic therapy trials will initially be focused on those ineligible for modulators, which represents a relatively small proportion of our patient population. And this is when we can start to lean on our study designs. Smaller population size, many trials. This is our call for innovation. Well, fortunately, we have a toolkit of innovative trial designs that we have an invitation from our regulators to tap into. Now, it breaks my heart a little bit today, but I am not going to review all of these trial designs because I promise this is not a textbook talk. But I don't want to leave you hanging a little bit. There are, for our more innovative designs, many designs use data external to the trial to more efficiently assess clinical efficacy of a new therapy. And this is sort of the trick to keeping these trial designs smaller. And here's the even greater news. Every CF research participant is part of this magic. We are using every research contribution to support these innovative trial designs, from past CF studies to our CF registries. Let me walk you through an example. In 2004, this little one, Cole, was the first participant enrolled in our EPIC clinical trial, at the time evaluating optimal treatments for early Pseudomonas. Fast forward nearly 20 years later, Cole is now enjoying a paddleboarding trip down the river, and from what I hear from his parents, working hard in college. Fortunately for us, his data are also working hard, along with others from this trial, to develop methods supporting innovative trial designs. Essentially, these designs bolster our smaller trial populations with existing data, helping us to decrease the size of a placebo group, or in some cases, even the need for a placebo group. So back to that EPIC data, we found that replacing or augmenting the placebo group with EPIC controls resulted in a comparable treatment effect, in this example, estimating the efficacy of azithromycin and reducing the incidence of pulmonary exacerbations. This proof of concept work lends itself to another innovative study design in our toolkit. And for those of you who are really wondering, this is the foundation for a Bayesian dynamic borrowing design that I just spared you a tremendous amount of statistical detail on. But I hope that you will see that the cumulative contributions of research participation are immeasurable when it comes to our innovative trial design strategy. These are those building blocks for our CF community that Trey just spoke of. Safety, however, is an even greater priority. Our Data Safety Monitoring Board oversees all of our trials and is comprised of a multidisciplinary team, including 20 patient representatives that provide thoughtful feedback on risk versus benefit of these therapies. Long-term safety follow-up studies will be required after every genetic therapy trial. This is unique to this class of therapies and will help us build confidence in the safety of these therapies over time. An important design consideration, however, is that co-enrollment opportunities in, for instance, symptomatic therapies trials are available during this long-term follow-up to maximize the shots on goal for an individual trial participant. Because remember, this is not a one trial at a time approach. And shots on goal are what we have so many of, as we just heard from Dr. Polineni, whether it be CF-related diabetes, NTM, or Pseudomonas. I want to be clear here that I am not the expert to talk about this multi-system disease. So I would like to introduce you to one of our experts, Kaylee, who on a more personal note has inspired me to keep focused on our mission since she was just a young girl.
In 2016, I graduated from my software development bootcamp, which was a huge accomplishment. I was very proud of myself. But shortly after graduation, I was hospitalized with kind of the run-of-the-mill CF exacerbation. It quickly turned into like the worst exacerbation that I've ever had. My doctor, who had been my doctor for probably 15 years by that point, sat right at my bedside and broke down, started crying, and told me that he thought it was time to start thinking about transplant. I was 20 when this happened, I think. I remember just that feeling of kind of hopelessness and frustration just being so amplified after that, because I just got like this bombshell dropped on me. And that was kind of the first of two really hard conversations like that I had to have. In the 3 years following graduation, I had a lot of kind of ups and downs with my health. Continually saw my lung function to decrease, over time, despite my best efforts. Just the main focus of every day was just keeping myself alive. Little did I know Trikafta would come into the picture. One thing kind of led to another, and that was like 2 months before I met Bo, I think. That was the thing that gave me the confidence and the ability to be able to consider dating. Trikafta has been incredible and has undeniably changed my life and saved me from transplant. Now that we kind of have modulators in the picture, clinical trials for the complications of CF, those are super important because they're all a step in the right direction towards managing this beast of a disease that affects so much more than just the lungs. I do still experience exacerbations from time to time, still intense IV antibiotics that have not so great side effects. Inflammation and kind of shortness of breath, CF-related diabetes and sinus symptoms and DIOS. That's kind of been a big one for me. You know, Trikafta is not a cure. I expect to eventually start to decline some more. I'm not naive to that. I've always been very interested in participating in clinical trials. Have never actually been able to participate. Either my lung function was too low or I was on antibiotics at the time, things that would automatically disqualify me. Hopefully someday I'll be able to. There's almost an element of paying my dues, I guess, in the community, kind of like helping advance these medications through the pipeline. Every year on my birthday, that's what I wish for, is a cure. It would mean a new lease on life. It would mean all of a sudden not having to worry for another minute about my future. Yeah, it would mean very grateful, triumphant, hopeful outlook on the rest of my life.
Kaylee and Trey articulated so well that there is still great need for new symptomatic therapies across our entire CF population, whether you are on or off a modulator therapy. At the same time, we must also acknowledge that many are undergoing transformational changes in their disease as a result of Trikafta. As we refer back to the PROMISE data, we see that in this cohort, the average FEV1% predicted 6 months post-Trikafta was near 91%. And this cohort also experienced significant decreased symptoms, with 32% having a CFQR respiratory domain score of the maximal possible score of 100, representing minimal symptoms post-Trikafta. As a result, trial design considerations will therefore be different between those with and without modulators. When developing a new symptomatic therapy in the CF population without highly effective modulators, larger treatment effects are expected in the absence of modulators, but we will still need innovative trial designs needed to move forward multiple therapies at the same time in the smaller population, as we have a very full pipeline. However, what's slightly different in this scenario is that we may not need to lean on those designs as much because we are inclusively developing these symptomatic therapies in the CF population with highly effective modulators. It's just that the trial design considerations are slightly different here. Smaller, measurable treatment effects are expected in the presence of modulators and more mild disease. It's not that clinical efficacy doesn't exist, it's just that it may be more difficult to measure with our traditional clinical efficacy endpoints. Thus, more sensitive endpoints are needed to detect the clinical efficacy of a new therapy. Let me explain this a little bit further. In this picture here, we see that there were dramatic reductions in the pulmonary exacerbation rate in the presence of Trikafta, as seen in the phase 3 studies, with even late-breaking data coming out of this conference to show that these rates could be even lower with longer-term follow-up. Even if we step back and assume these slightly higher rates, we would need a sample size of over 1,000 participants to detect a meaningful reduction in exacerbations in a future trial. Now, I learned my lesson in sample size 20 years ago, and we're going to need a different strategy. We also don't want to go back to a one trial at a time approach with our very full pipeline. So how are we going to do this? Well, to date, all of our successful therapies have relied very heavily on clinical trial endpoints such as FEV1 and pulmonary exacerbations to detect the clinical efficacy of a new therapy. Well, this is now a moving target as the disease is literally changing before our eyes. We have several studies ongoing that are mapping the outcomes of people with CF on Trikafta, but the story is still unfolding. And so while these studies are still ongoing, we will still need a strategy for our clinical trials. And this is one where, you know what, it is okay sometimes to bend the rule book a little bit because we cannot afford to miss detecting clinical efficacy of a new therapy. Fortunately, we have many endpoints to aim here. We have imaging and lung clearance index, which may more sensitively help us detect changes in the lung in response to a new therapy. As you see here in the CT scan, which can detect bronchiectasis and mucus plugging despite a normal FEV % predicted. We also have new patient-reported outcomes being developed. The PROMS project is being led by our colleagues in the European Clinical Trials Network and is modernizing our patient-reported outcomes with direct patient involvement, capturing how patients feel, function, and survive amidst this changing disease. This project is complemented by the core outcome set task force being led by researchers in the UK and is combining and unifying the experiences of our CF community, researchers, and regulators to create a core set of outcome measures that can be used across our clinical trials and reflect meaningful outcomes across this multi-system disease, not only focusing on those respiratory endpoints, but for instance, including GI symptoms. It is projects like these that will be critical to help us define what belongs in that new target circle for clinical efficacy. But it is not just meaningful endpoints that we need as part of our clinical trial strategy. We also need innovative endpoints. We have learned what we can accomplish from the comforts of our own home with a little bit of technology. And this has inspired the Outreach study, which is optimizing and evaluating the feasibility of remote spirometry as a clinical trial endpoint. Importantly, this study could not have been envisioned without direct involvement from CF community members to ensure that all of that collective experience with those remote spirometers were incorporated into the design of this study. And it is in fact the validation of remote endpoints that are the gateway for what we call decentralized trials. These trials use telehealth and remote endpoints to augment or even replace in-person study visits. This is just another design in our growing toolbox with a goal towards increasing accessibility to trial participants, and yet another way we can get towards those larger trials. Now, I've spoken a lot today about new trial designs and new endpoints, but ultimately, we must keep in mind that we are trying to get our therapies from clinical trials all the way to regulatory approval to get into the hands of our patients. And it's not just one regulator that we see here, but there are multiple regulators across the globe that we must satisfy with our approach. This is really one of the critical focus areas of the global partnership across our clinical trial networks, working together to build harmonized roadmaps to ensure that we are not duplicating efforts. We need to ensure that clinical efficacy and safety data that are generated in one country can be used to support regulatory approval in another country, because that is what is going to get us faster through this pipeline. At the same time, we humbly acknowledge the increased partnerships we will need between our industry sponsors and reimbursement bodies to overcome the global disparities in access as these therapies are rolled out to our global community. Our trial pipeline is working so hard to ensure that everyone will reach their moonshot. I just want to take a minute to reflect again on the progress that we have made. In 2016, Dr. Patrick Flume stood on the stage and hinted that we were getting close, and that was just prior to the time of Trikafta being on the horizon. We are in fact moving closer, as we see with the striking data from the CHECK-SC study that shows that nearly 80% of a large cohort on Trikafta now have a sweat chloride value below the diagnostic threshold of 60. These data are absolutely breathtaking. At the same time, we see that there is some individuals that we still have potential to move below this threshold with perhaps our newer modulators. And also, I just want to take time to reflect that each and every data point on this plot reflects someone who is undergoing a unique experience with Trikafta that we must continue to listen to and support. At the same time, these data just provide collective motivation still that we need to make room in our pipeline to understand the new priorities of our CF community. And through the important work of the James Lind Alliance and the CF Community Voice, we have heard loud and clear that the #1 priority from our community is to study the effective ways of simplifying treatment burden. So yes, we are progressing a pipeline of new and novel therapies. At the same time, we are now studying whether some therapies can be simplified or reduced. Just let that sink in for a moment. I can assure you it is a scenario that was never covered in my book. It is a great sign of progress. And the first question that has risen to the top is, what is the impact of discontinuing inhaled therapies after Trikafta? As you can see here, there is no one strategy that can meet the needs of all of our stakeholders. And our pipeline has delivered. We have randomized trials to collect both short-term and long-term outcomes. We have a real-world observational study and a patient perspective study to show us how we can design these studies even better in the future. And what isn't so clear is that when we partner with the community to address their priorities, what we can accomplish is absolutely magical. Nearly 2,000 participants have enrolled across these trials in the last 2 years. This is unbelievable commitment from our trial participants, our coordinating centers, and our research sites during a time that has been professionally and personally challenging for so many during the COVID-19 pandemic. Simplify is the first trial to complete in this pipeline and enrolled people with CF 12 and older established on Trikafta. Participants were randomized to one of 2 trials to either continue or discontinue either hypertonic saline or Dornase alpha for 6 weeks. I am privileged, on behalf of my co-PIs, Drs. Alex Gifford and David Nichols, to present the results of this study today, which were just published about an hour ago in Lancet Respiratory. Among those 12 and older with relatively good lung function on Trikafta, in fact, this cohort had an average baseline FEV of 97%, which is remarkable. Discontinuing daily use of either hypertonic saline or Dornase alpha did not result in a meaningful change in lung function. As we look at this figure, we see that the treatment effect estimates for both trials were near zero, minus 0.32% in the hypertonic saline trial and 0.35% in the Dornase alpha trial, with the lower bounds in the 95% confidence intervals excluding our predefined non-inferiority margin of minus 3%. We also measured that more sensitive endpoint of lung clearance index and found no clinically meaningful changes in LCI occurred in either trial, with again the treatment effect estimates near zero and the confidence intervals excluding the minimal clinically important difference as indicated by the SHIP-CT study, which evaluated the efficacy of hypertonic saline in young children with CF. Interestingly, in a small sub-study, mucociliary clearance significantly improved in the discontinuation arm as compared to the continuation arm in the Dornase alpha trial. Few participants who discontinued experienced more respiratory symptoms, and no meaningful treatment differences were seen in other key secondary outcome measures. Overall, I have to say, I never thought I'd be so excited to stand on the stage and report study results that showed no treatment effects. These data represent a potential for a paradigm shift in clinical care, and our community has been working and waiting for a long time for these results. However, there are some important caveats. Simplify included people on Trikafta 12 and older with, on average, very high lung function. Thus, the results may not predict what would occur in people dissimilar to this group. Also, respiratory adverse event data highlighted a potential caution for those with lower lung function. This was a very small subgroup in our study and deserves further evaluation. Simplify was a 6-week trial. It was not designed to evaluate long-term outcomes. However, forthcoming studies, including CF Storm and Hero 2, are evaluating longer-term outcomes over a 12-month period and will provide additional data to enable shared decision-making between people with CF and their care providers regarding treatment discontinuation. And with every study, there's even more questions and more priorities, including the need to evaluate the effects of altering other foundational standard of care therapies in the presence of Trikafta, such as airway clearance therapy or inhaled antibiotics that were not evaluated in the context of these studies. Also, we need to better understand the timing and necessity of starting certain daily therapies early in life, as Trikafta is rolled down to younger pediatric populations. And so we come full circle today, with many priorities, many trials, and a full pipeline. It will take everyone to ensure that every individual with CF will reach their moonshot. Thank you to everyone who makes our clinical trials possible. This moment does belong to our trial participants, as we thank you for your time, voice, sacrifice, and commitment. And on behalf of Dr. Polineni and I, we just want to thank Dr. J.P. Clancy and the CF Foundation again for the opportunity to be here. And I just want to give a personal thank you to one of my colleagues, Dr. Christopher Goss, who I have the privilege of working with every day at the Coordinating Center. And again, Dr. Bonnie Ramsey, for giving so many of us the opportunity to do this work. And we give our commitment to you that we will not stop until it's done.